we are working on the human trophoblast stem cells derived EVs or exosomes. Today, I'm going to elaborate a little bit more on the human trophoblast stem cell secretome that we use in order to isolate the exosomes or extracellular vesicles and treat the senescent cells with these. So imagine a world with the abilities to delay age-related diseases, extend healthy lifespan, and prevent maybe age-related diseases. That would be wonderful. And that's what we're actually trying to do here at the National Institute on Aging. As a brief introduction about aging, these are basically the hallmarks of aging. And here I would like to focus on senescence, which is one of the major hallmarks of aging. It just also happened that we have the stem cell exhaustion just nearby senescence. And in my interest, I'm actually interested in understanding both the senescence and stem cell exhaustion in aging and how to prevent these two phenotypes in accumulating with age. As you know, senescent cells, they have some certain characteristics. With age of the cell, it actually gradually accumulates DNA damage, which causes a growth arrest. And sometimes we feel like this growth arrest is reversible or reversible. It depends on the intervention. And maybe it is reversible in the future. We can find some programs that would help us to reverse the senescence phenotype. One of the main characteristics is that these cells are actually resistant to cell death. So one of the major issues is that you try to kill these cells or try to eliminate them, but they are resistant to cell death. One major issue also is that these cells they have very active metabolic system as they keep secreting senescence-associated secretory phenotype, or they are associated with this phenotype that promotes inflammation as major components of the senescence-associated secretory phenotype, or SASP. These are pro-inflammatory cytokines. And one major issue with senescence cells, since they are not dying very easily, and also with age, we have a declining immune system to eliminate these cells from our tissues and organs. They tend to accumulate as we age. And that's a major issue because these cells are now contribute into very many age-related diseases. And that is why in our lab and many different labs, we are trying to either eliminate these cells or inhibit their effects on the microenvironment. Approaches that are currently being very active or actively pursued by so many different labs would include parabiosis, in which you connect the circulating system from the old and young mouse, and that turned to be very beneficial from the old animals in terms of rejuvenating the organs and preventing age-related diseases. Another approach that we're very familiar with is the caloric restriction, or to have some nutritional interventions. And these also work very well in somehow reducing the burden of age-related diseases. Well, as Joanna mentioned in her presentation, reprogramming is a very important direction to try to prevent age-related diseases or reverse the phenotype that might be associated with the stem cell exhaustions. We have also other labs that do partial reprogramming or full reprogramming using the hardcore factors, including the old Yamanaka factors. Another approach that we are also working on is xenotherapy. First one is senolysis, which directly eliminates senescent cells, but we also have xenomorphics that do prevent the impact or the release of pro-inflammatory cytokines that are normally secreted by senescent cells. So with these, since the parabiosis work, so we asked ourselves if we can instead of connecting the two animals' circulating system, we can try to treat senescent cells with the stem cell-derived exosomes or extracellular vesicles, EVs, and non-EV particles. So the secretomes that are coming from the conditioned media cultured with the stem cells could actually be somehow preventing or somehow counteract the impact or the released phenotypes or the released cytokines from the senescent cells. So we asked this question, but to me it was very important to ask which kind of stem cells shall I use. That's why I searched for the earliest stem cell possible, and I found the human trophoblast stem cells from Accelerated Bioscience to be the most suitable and earliest stem cell that I can use for this purpose. And that's why I contact Yota Ali, the founder of Accelerated Bioscience, and he explained that the human trophoblast stem cells that they are isolated from ectopic pregnancy, and they have no ethical issues with that. And with us working at the National Institute of Aging, we do care about the ethical issues very much. So that that gave me the green light to contact the Yotali and the Accelerated Bioscience so that they can provide the secretome from the human trophoblast stem cells. The fact that they are isolated or outsourced from the ectopic pregnancy, failed pregnancy, that is very important to us, and also represents a very early stem cells, which could be very powerful in case that we want to prevent the age-related diseases. So as soon as we received the material, the secretome from Yotali and Accelerated Bioscience, we had already a plan in place. First, we wanted to study the effect of the secretome from the human trophoblast stem cells conditioned media. We wanted to study the effect on the recipient cells. And here we use the WI38 cells. These are human diploid fibroblasts that are commonly used to study senescence. And the second part is to characterize and find out or explore what does really exist in the secretome from the human trophoblast stem cells when it comes to exosomes or the non-EV particles that do also exist in the secretome from the human trophoblast stem cells. The third part is to identify the impact or the effect of the human trophoblast stem cell derived secretome on aging or age-related traits, including inflammation and other age-related traits. So first here, we treated the human diploid fibroblast WI38 cells with ionizing radiation or etoposide to induce senescence. And simultaneously, we treated the cells with the non-conditioned media or the human trophoblast stem cell conditioned media or the secretome. And we harvest the cells at day 5, and we looked at the expression of some pro-inflammatory cytokine inside the recipient cells at the mRNA level. And as you can see here, the transcripts that are encoding for IL-6, CXL1, ICAM1, and GDF15, these are all aging markers. We find that the human trophoblast stem cell-derived conditioned media or the secretome can suppress the expression of these mRNAs. The same we observed when we induced senescence using etoposide. And with that, we wanted to do global analysis. As we observed the reduction in these mRNAs, so we sent the mRNA for RNA sequencing to assess the global transcriptome changes in cells that are treated with the secretome. And as you can see here, a lot of pro-inflammatory cytokines or SASP factors, we found that they are downregulated by the human trophoblast stem cell derived secretome, including MMP1, GDF15, CXL1, and so many on the left side here in the volcano plot. You can see that they're all downregulated. All of these are important cytokine, chemokine, metallopeptidases, and they're involved in many in the SASP phenotype or SASP pathways as well. So these are at the transcriptome level. So we wanted to see what happened at the secretome level of the recipient cells. To do that, we performed all-link proteomics. And as you can see here, the same treatment, we collected the secretome from the recipient cells. So don't confuse that. This is the secretome now from the recipient cells after the treatment with the conditioned media from the human trophoblast stem cells. As you can see here, all of these red color, all the secretome, all the secreted proteins, a lot of secreted proteins are released into the media in the case of non-conditioned media. However, when we treat the cells with the human trophoblast stem cell condition media, or the secretome, we observe dramatic downregulation of the secretome of those cells that are treated with ionizing radiation, indicating that the human trophoblast stem cell secretome and possibly the exosomes also can suppress the production and the secretion of the pro-inflammatory cytokines. And these include, as I said, IL-6, GDF-15, and so many different other pro-inflammatory cytokines, indicating that there is a suppressing effect. So the human trophoblast stem cell condition media can actually suppress SASP factors if you induce senescence with the ionizing radiation. So with that, we also wanted to validate these at an individual cytokines or individual SASP factors. So with the bioplex, or this is kind of ELISA assay, we also looked at IL-6, IL-8, GDF-15, and as you can see here, these are dramatically downregulated when cells are treated with the human trophoblast stem cell secretome. And we validated so many different ones. Just for the sake of time here, I'm showing IL-6, IL-8, and GDF-15, and so MMP3 as well, and ICAM1 and osipontin. So here we can conclude that the human trophoblast stem cell condition media, or the secretome, can actually reduce the secretion, the production of these pro-inflammatory cytokine, and as well as the secretion of this one to the microenvironment. So next, we shift the gear to the exosomes, or the extracellular vesicles. So we use these, the secretome, we from extracellular bioscience, and we use these the size exclusion chromatography to isolate and purify the exosomes or extracellular vesicles from the condition medium. And as you can see here, we could successfully isolate and purify the exosomes from the condition media. And this is an evidence here, as you can see with the electron microscopy, we can see it's very nice, pure. We counted the particles, and then we started to treat our cells with these exosomes. And as you can see here, first we wanted to confirm the internalization of these exosomes. We labeled the exosomes with the fluorescent dye and then treated the cells with these exosomes. And as you can see here, we can successfully detect these exosomes inside the cells, indicating that there's no problem with the uptake or internalization of these exosomes. And then we repeated the treatment again with the ionizing radiation, but in this case, we added the exosomes. And day 1 and day 3, and then we harvest the cells at day 5 to do the RT-qPCR, real-time PCR analysis, to assess the levels of IL-8, 6, L1, GDF-15, and other pro-inflammatory cytokine, including mRNAs. And as you can see here, very nice, very significant downregulation of these pro-inflammatory cytokine, including mRNAs. So here we understand now that the secretome, including the exosomes, can actually suppress the SASP factors when we induce senescence either by ionizing radiation or etoposide. But the main question here is, how does that happen? So this is very important to us at the molecular level. We would like really to assess the mechanism that mediates the effect of this suppressive effect on the SASP factors. To do that, we started to look at the characteristics, what is really in there in the trophoblast stem cell derived exosomes, or the proteins that are secreted by these stem cells. So to do that, we started with the O-link proteomics, simply by taking the non-conditioned media compared to the conditioned media, or the media that is coming from the human trophoblast stem cells. And as you can see here, this analysis revealed a significant number of proteins that are secreted by the trophoblast stem cells, indicating that this is a rich environment that might contribute into the suppression of pro-inflammatory cytokines in cell culture that we observe. But very important to us is to confirm where these material are coming from. And as you can see here, very important to us to look at the cell marker to confirm or to see what kind of material do we have. And surprisingly, we saw a broad stem cell signature across lineages. You can even see mesenchymal stem cells here from different organs, from liver, from bone marrow, from adipose tissue, indicating that this is not only a stemness signature of the human trophoblast stem cells, but also represent a broad stem cell signature across different lineages. So with that, we feel comfortable that we are indeed working with pure material that traces back the origin of these cells, and we can now characterize those proteins. We see multiple proteins are significantly enriched in the human trophoblast stem cell secretome, including pentraxin 3 and also server collagen proteins that are also existing in or rich in the trophoblast stem cell-derived secretome or condition media. Collectively, these factors that are secreted by human trophoblast stem cells, they are pro-growth, they are pro-repair. So you can see there are some factors that are pro-growth and also demodulate the extracellular matrix, and these are signaling proteins also that can promote the changes in either cell growth or extracellular matrix remodeling or cell adhesion. So we also see some DNA repair proteins. So generally, this kind of environment is kind of promoting the growth and repair mechanisms that might be happening after induction of senescence using ionizing radiation or etoposide. So remarkably, we also here isolated the exosomes and performed mass spec analysis, and we detected large number of proteins. But however, for me, it was interesting to see that there is a significant number of proteins that are like 130 proteins. These 130 proteins that are normally known to be downregulated in senescence. So that is kind of restoration here. So there might be some potential restoration in this secretome that is coming from the exosomes or coming from the secretome itself. That might be including some particles and nanoparticles, soluble proteins. But in addition to that, the exosomes themselves are bringing other repair mechanisms or supporting mechanisms that are somehow potentially maybe restoring the missing proteins that are otherwise downregulated by senescence. So these are in blue color, these are the proteins that are downregulated in senescence, and these are the proteins in pink here, 130 proteins that are coming from the exosomes released by the human trophoblast stem cells. So propose there might be some potential restoration of key proteins that are involved in key processing, including translation, RNA processing, and the extracellular matrix, DNA repair. So this is kind of cocktail, like a magic cocktail that might be actually rejuvenating the cells or preventing some detrimental phenotypes that are induced by senescence. In addition to that, we wanted to see major pathways, including the DNA damage and NF-κB pathway. And as you can see here, when cells are treated with the ionizing radiation to induce senescence followed by non-conditioned media, you can see in COMET assay a large tail of DNA, indicating that these cells have DNA damage. However, when we add or when we treat the cells with the human trophoblast stem cell secretome or conditioned media, we see a significant reduction in DNA damage. So there might be some protection against a DNA damage or some acceleration of DNA damage repair by the conditioned media. And this is the quantification indicating that is kind of significant. When we add the secretome, after inducing senescence in WI38 cells, we observe a significant reduction in gamma H2X, which is a marker for DNA damage. And also, we observe a downstream effect on that, which could be the phosphorylation of NF-κB, which is a master regulator of SASP factors. So with the conditioned media and also with the EVs, we observe similar pattern, indicating that maybe the secretome is also suppressing, or the EVs are also suppressing DNA damage or promoting DNA damage repair, at least in part by the existing exosomes in the secretome of the human trophoblast stem cells. So the last part that we wanted to address is what would be the effect of secretome on the aging phenotypes. To do that, we injected the control animals with the non-conditioned media, also injected the human trophoblast derived conditioned media or the secretome. We injected them in aged mice, and we looked at circulating biomarkers of aging and inflammation. As you can see here, we could observe a downregulation of IL-6 in the blood, CXL1, BAF, and DPP4. And we are also trying to reproduce these results in a larger cohort at this point. And maybe also as soon as we have those, we can do a larger, like more comprehensive analysis using the Olink inflammation panels. So I hope I could convince you that the human trophoblast stem cell secretome is kind of rich in its contents, whether it is the exosomes or the non-exosomal particles in the secretome of these cells. And somehow these cells, we still don't know the exact mechanism, but we see suppression of DNA damage, or we see accelerated DNA damage repair, which subsequently can have some impact on the activation of NF-κB, which is a master regulator of pro-inflammatory cytokine like IL-6, IL-8, and GDF-15, and so on. So in the future, our perspectives now are basically to target age-related diseases like Alzheimer and other diseases like diabetes, and also try to investigate the effect of the secretome and the exosomes on stem cell exhaustions. So hopefully in the new studies that we will try to do using the exosomes is to investigate the anti-aging effects, inflammation, tissue regeneration, neurodegenerative disease like Alzheimer, and also any other age-related disease that we can see the impact of the exosomes or the secretome on the aging phenotype. With that, I would like to thank you all. Thank you very much for this, for organizing the forum. I would like to thank Miriam for giving us the opportunity to do this in her lab. Martina Rossi worked a lot on these, and also the Lab of Clinical Investigation, Dimitri Kaprianis, a lot of experience, and also Yota Lee, and I'm happy to receive any questions.